Whoa. This is protein synthesis, a doozy. So again, y'all, we all know that DNA stores the genetic information. And they're stored as genes, which are sequences of DNA nucleotides that can be expressed to produce a final functional product, either a polypeptide or an RNA molecule. And so we have this concept known as the central dogma of life. We know that, again, the flow of genetic information goes from DNA to RNA, specifically M, and then using M, R, and tRNAs to produce the protein. And again, the central dogma of molecular biology. So, in these steps, y'all, from DNA to mRNA, we have transcription. This process occurs in the nucleus or in the cytoplasm of prokaryotes. Then we have translation, where we have that mRNA being used as a template to produce a protein. Okay, y'all. If you guys can't remember which one goes first, always use the alphabet. C goes before L. So transcription before translation. So the first process is transcription, in which, again, the mRNA is created inside the nucleus from a DNA template strand for eukaryotes. Okay, so what does this, y'all? Well, RNA polymerase. Just like with DNA synthesis or DNA replication, this transcription process requires enzymes to help speed this process up. So this RNA polymerase has two jobs. The first job is to unwind the DNA, and then number two, to add the nucleotides to create the mRNA. So the RNA synthesis follows the same base pairing rules as DNA, except that uracil is substituted for thymine. So we can see that process and the role of RNA polymerase in this picture here. It attaches to the DNA and then unwinds it, and then again starts adding the RNA nucleotides to create the mRNA. And notice that it creates in the 5' to 3' direction. So meaning that nucleotides are being added on at the 3' end. Okay? So once it's complete, the RNA polymerase leaves, and we have our RNA mRNA. In both prokaryotes and eukaryotes, there's a promoter area. This area is where RNA polymerase attaches to, and this promoter, noted, is on the DNA. But specifically, in eukaryotes, y'all, the promoter area contains something called the Tata box. So it's just a short segment of AT basis pairs that is important in forming the initiation complex in eukaryotes. So proteins called transcription factors help binds RNA polymerase to the promoter area. And once it is done, once the RNA polymerase binds to the promoter area with the help of transcription factors, this whole thing is called the transcription initiation complex. As stated before, RNA polymerase has two jobs: unwinds the DNA and then builds in the 5' to 3' direction. Okay, it reads in the 3' to 5'. So we can have a general statement here, and it applies both DNA and RNA polymerase. The polymerase reads from 3' to 5', and then builds in 5' to 3'. So good general rule. And this process continues until we see the termination sequence. That signals the RNA polymerase to detach and that the gene for that specific protein is complete. Whenever a gene exists on a DNA molecule, one strand is the coding strand and the other is the non-coding. So this concept is a little confusing if we are taking it literally. So let's take a look at the strand type for transcription. So the coding strand, also called the sense or non-template, the non-template should be the indicator here, matches the RNA transcript, meaning matches the mRNA being coded. So the coding strand is not being read by RNA polymerase. Okay, so again, let's take a look at that specifically. We have 5 prime, 3 prime. This is 5 prime to 3 prime. And so this right here must be our coding because we have AUG, ATG, and then GCA, GCA, and it matches exactly as the mRNA being created, except that the T is a U. Now, the one that is being read is the non-coding strand, or the template strand, sometimes called the antisense. So this one is being read by RNA polymerase, as seen in the image. Okay, so non-coding or template strand. So this process only occurs in eukaryotes. The mRNA that's being produced in prokaryotes is already being translated at the same time. We'll get to that later. But in eukaryotes, y'all, the mRNA that's made directly from transcription is known as pre-mRNA. The pre-mRNA must be modified before leaving the nucleus. The first thing that happens, again, is that the 5' end receives something called the GTP cap. This is simply similar to ATP, but instead of an A, it is a G. So the other end is the 3' end, which is a poly A tail. So this poly A tail has many A's, similar to the sophomore Aggie yell, A. So again, if you need to remember, it's the poly A tail, right? Now, these modifications again has a purpose. The first one is to protect from hydrolytic enzymes. The other two, again, facilitates the export of the mRNA into the cytoplasm and then help the ribosomes attach to the 5' end of the mRNA. And then the genes, y'all, from eukaryotes have long non-coding regions called introns. And then they lie between the coding regions. The coding regions, the one that expresses the protein, is the exons, because again, they are expressed and translated into amino acid sequences. So what must be done before the mRNA can leave the nucleus, and again, only in eukaryotes, is that we have to splice the RNA. This process is called, again, RNA splicing, which removes the intron and then joins the exons. So let's talk about this and how we could remember it easier. So for the introns, the introns, right, are like intruders of the code. And so what do we do to intruders inside the house? We remove them. So again, the RNA splicing process removes the introns. Then we have the axons. The axons are express. So the EX would be the express. That is the one that is creating the desired protein. So prokaryotic genes do not contain introns and their mRNAs are not spliced. So what does the splicing in eukaryotes? An enzyme called spliceosomes. So once the mRNAs has finished their modification, it then goes and leaves the nucleus. So right now, the mRNA has sequences of nucleotides that calls for a specific amino acid. So these codes are read in triplets. Again, mentioned before, codons. They're three nucleotides in a row, and they call for specific amino acids. There are a total of 64 possible codons, but only 20 amino acids. So obviously, these codons will call for the same amino acids um, some of the times. Okay, so for most amino acids, there's more than one codon, and the genetic code is considered to be redundant. But it's not ambiguous because each codon specifies only one amino acid. Let's look at that. So again, the genetic code is nearly universal. The codons that specify amino acids are the same in all organisms. The genetic code is ancient and it unifies life and indicates that we come from a common ancestor. Okay, so this allows us to genetically modify organisms, thus basis for genetic engineering. And you guys can see that table here. The codons call for the same serine. Again, these codons call for threonine. And then we have these guys for proline. So again, redundant, but not ambiguous because they're very specific. So again, let's use an example of how to read the codon chart just in case you missed the other video. We have the codon A. So how to read it? We go all the way to the far left. I identify A. So this is the region we're going to concentrate on. Ignore everything else. That's the first letter. Second letter, we have the A. So we have to look for this, move down. And so is this the box that we're going to concentrate on? And finally, we have our last A. So we take a look in the third, far right, third base area, and we circle and move. And it's this line here, A. It is lysine. So the amino acid attached or calling for, being called for AAA is lysine. So once the mRNA is in the cytoplasm, it goes and find a ribosome. Okay, so the ribosome is the site of translation. So during translation, sequence of nucleotides in an mRNA strand is converted into a chain of amino acids with three main steps: initiation, elongation, and termination. So we see the initiation process here. Translation starts when our RNA in the ribosome interacts with the mRNA at the start codon. So this mRNA is attached to the small subunit, reads it until it gets to the AUG. Then we have the large subunit binding to it. And the second code is being read. And again, that amino acid attaches to the arriving amino acid, and the bond here breaks. So now this tRNA can leave as seen here to get another methionine. Okay, so that is initiation. So the process starts and it builds the amino acid sequence. So we have the ribosome reading in the 5' to 3' direction. Okay? Then the tRNAs have the anticodon that's complementary to the codon on the mRNA strand. And so we see that there, complementary. So again, this direction we see is 5' to 3' direction. So it's moving in this direction here. Okay, so being red, and then the mRNA is moving this way. So this must be the 3' to 5', 3' to 5', 3' to 5'. Okay, let's continue. So each tRNA carries a specific amino acid, ensuring that the correct amino acids are brought as each codon is read by the ribosome. So that process is here. The codon, again, is ready. tRNA anticodon attaches to that. And we see here that, again, this bond is broken and transferred over to the new one. And so once that is transferred, this tRNA is going to move to this area here and leave the ribosome. And the process continues in that process. So the process that you saw earlier where the existing tRNA cuts off the bond and transfers to the new one keeps going until a stop codon is reached. That is when a release factor is attached to this codon, and then it causes the tRNA to leave, detach its polypeptide, and then the large subunit, small subunit, and mRNA all disassemble. So we ask, hey, what happens to that mRNA? Well, that mRNA can go find another small subunit and get translated again, or over time, parts of the mRNA is broken down even with that 5' cap and then 3' tail. So let's compare the process between prokaryotes and eukaryotes. The biggest difference, y'all, is that prokaryotes, they have their translation and transcription happening at the same time. So again, as this mRNA is being created, which is transcription, this right here, the proteins are being made, translation. It happens at the same time. So the nuclear envelope, however, separates transcription from translation in eukaryotes, right? And then that pre-mRNA in eukaryotes must be modified first, the GTP cap, the poly A tail, and then the removal of the introns must all occur in the nucleus before being translated in eukaryotes. mRNA is not modified whatsoever in prokaryotes. Big difference. So, big central dogma of life, y'all. Again, we have the flow of genetic information, DNA to mRNA, and this is transcription. In eukaryotes, the mRNA must be modified first, the GTP cap, the poly A tail, introns remove, exons join, and finally leaves the cytoplasm to be translated. So again, this happens at the ribosome, and then again, the protein is made. So let's take a look for the summary right here. This is obviously for a eukaryotic organism because the nucleus is present. We have pre-mRNA being made. We have the RNA being processed. We have the GTP cap, poly A tail. The introns are going to be removed right here. The cap helps with the attaching of the small subunit of the ribosome. Once AUG is read, the start codon, the tRNA is going to be attached to it, and then the process of building the amino acid continues with this bond being attached to the arriving amino acid, and then it leaves. This happens until the release factor is attached to the stop codon, and everything disassembles as you see right here. Okay, so that is it. Watch for a review overview where I'll be drawing the process of protein synthesis from start to end, and then I'll have it summarized for y'all in 2 slides. So again, have a good one.